Hello everybody and welcome to this module on telomerase repeated ampli protocol, or TRAP for short. So let me begin by discussing some important terminology. I'll be discussing non-telomeric oligonucleotides. These are known as telomerase substrate, or simply TS primer for short. So you have to keep this in mind throughout this module. Let me begin by discussing the principle of TRAP. The main function of TRAP is to determine telomerase activity. Here to the right, you can see a figure and you can see a gel electrophoresis of TRAP products. These are the bands that you can expect with the conduction of a TRAP experiment. And there are 3 steps to telomerase repeated amplification protocol. The first one is extension or elongation. The second step is amplification, and the third step is detection. Generally speaking, detection is performed by gel electrophoresis, specifically polyacrylamide gel electrophoresis. Now let me talk about the first step, which is extension or elongation of the telomerase substrate. So telomerase is used to add telomeric repeats to the telomerase substrate. Remember, the telomerase substrate is also known as the non-telomeric oligonucleotide. That's because the telomerase substrate is a non-telomeric oligonucleotide. It's in the name. So the telomerase substrate is a primer as well. So it's used as the forward primer as well. And so here you can see a figure, and you can see here that the telomerase substrate is being extended. The extension is done by the telomerase enzyme, and you can see in italics the extension of the telomerase substrate. Now, after the extension of the telomerase substrate, we'll use a reverse primer. This is called the CX primer, and it has some mismatches in it. So you can see a mismatch here. You can see a mismatch here. They're labeled in asterisk. You can see a mismatch here, and those mismatches are formed or made in primer design so that we can prevent primer dimer formations. So let me talk about the amplification step. In the amplification, we are amplifying the telomerase synthesized DNA. And so what you have to keep in mind here is that extension products are amplified by PCR, and we'll use a reverse primer, which will anneal first, followed by a fluorescent TS primer. So you use a reverse primer and a fluorescent TS primer. Generally speaking, the TS primer used first in the extension step by telomerase is not a fluorescent primer. Now, keep in mind, you can also use a radio-labeled TS primer. So radio-labeled with P32, for example, or 32P. And so the reverse primer here, as I said earlier, entails mismatches. This reduces primer dimer formations. And you also have a fluorescent TS primer that's also going to be used in the PCR step. So once you've done the first initial extension reaction of the telomerase substrate, or the non-telomeric oligonucleotide, the next step is that the reverse primer will anneal to the extended product. So the extended product can be seen here. This is the extension shown in italics, and you can see the reverse primer annealing to it. You can also see the mismatches in asterisks. And what you have to keep in mind here is that the CX, or the reverse primer, will produce a second strand of DNA, and then you will perform a PCR amplification where a fluorescent TS primer is going to be used along with the same reverse primer that we use, the CX primer, to amplify the extended telomerase product, or the extended telomerase substrate product that has been extended by telomerase. There are various detection methods for TRAP. So here are detection methods that utilize gel electrophoresis. Specifically, we utilize polyacrylamide gel electrophoresis since it offers a higher resolution, which is important to show the processivity of the telomerase repeated amplification protocol product, or the TRAP products. So one option is to use a fluorescent TS primer. This is sufficient to show the TRAP products. Alternatively, and what originally was used, is a 32P radio-labeled TS primer, and then followed by autoradiography. In addition, you can also simply use cyber green staining in the gel. So this is a conventional in-gel staining that you can use to showcase the TRAP products in the gel electrophoresis. So in terms of gel electrophoresis, polyacrylamide gel electrophoresis is performed afterwards. This is used to detect the presence or the absence of the amplified products. What you have to keep in mind here is how the gel will look like and what it means in terms of its visualized look. So telomerase activity, when we talk about telomerase activity, we're talking about the ability of telomerase to synthesize or to extend the telomerase substrate. So it is proportional to the total amount of DNA that is synthesized. So you have to keep in mind that telomerase activity is proportional to the total amount of DNA that it synthesizes. When we talk about processivity, we are talking about the length of the fragments synthesized and the difference of those lengths as telomerase extends more and more of the telomerase substrate. So the telomerase processivity is proportional to the length of the fragments synthesized, and here processivity refers to the telomere or the telomerase ability to add multiple hexameric repeats in the case of vertebrates, which is TTA-GGG repeats, such as TTA-GGG repeats in vertebrates or mammalian cells. So this is important to understand when anticipating the results. So when we talk about processivity, we're talking about, in the case of vertebrates, the 6 base pair incremental ladder that forms. So you have an incremental ladder that is formed here, and it's a 6 base pair incremental ladder. And the reason that is because in the case of vertebrates, that is because you have TTA-GGG repeats. These are the telomeric repeats that are added by telomerase. When we talk about telomerase activity, on the other hand, we're talking about the total amount of DNA that telomerase is going to synthesize. So activity differs from processivity. Processivity refers to the ladder that is formed, whereas activity is referring to the total amount of DNA that the telomerase will synthesize. Let me talk about the methodology here of TRAP in more detail. So here you can see a non-isotopic detection, and you can see to the right an isotopic detection of telomerase activity by TRAP. Specifically, this comes from a particular kit for TRAP that you can visit on this link. And what you see here are the steps. Let's go over the steps for non-isotopic detection or non-radio-labeled telomerase. So the first step is to extract telomerase, after which you prepare a reaction mix for the telomerase extension. So the telomerase extension is usually done for 30 minutes at 30 degrees Celsius in mammalian cells. You then perform PCR amplification. This is 30 to 33 cycles. You can see PCR amplification for isotopic detection is a little bit less, since isotopic detection is easier or more apparent than non-isotopic detection. So you then use a polyacrylamide gel so that it is roughly 10% or 12.5% polyacrylamide gel electrophoresis. This is the same for non-isotopic and isotopic detection. This is so that you can offer the appropriate resolution to showcase the TRAP products. And then you can stain the gel, and you can perform gel photography or imaging and perform data analysis. In the case of isotopic detection, you do the same thing, except you use a dry gel and you use a autoradiography method so that you can then perform the data analysis after. Let me talk about further troubleshooting information that pertain to TRAP. And this also comes from the same link, from the same TRAP kit. It's also quite helpful. So if you see no visible products in any lanes, and this kit offers many different controls, but we have to keep in mind is that if you see no visible products, then several things could be problematic. The first thing is that the primers could be problematic. The DNTPs used could be problematic. It could be the Taq polymerase enzyme that's problematic. So you want to check all of that. You also want to check the lysis buffer. You want to check all the various components that are involved in the reaction. Make sure that all of them are have been appropriately. All the steps have been appropriately performed. The other thing you can check is check the thermocycler for the proper temperature and the time setting. So this is very important. PCR is very sensitive. However, you want to make sure that the appropriate temperatures have been used for TRAP, for the PCR step of TRAP. You can check the annealing temperature, and you can also check if Taq polymerase is active. Now, the other thing you can check is if you have few or no TRAP ladder bands are being visible. So if you have, say, poor processivity, then you can dilute the extract. You can dilute it by 5 times or 25 times or 125 folds with the lysis buffer that you use. In this case, in this kit, they use a CHAPS lysis buffer, and then reperform the PCR. this is probably the best thing that you can do. But in addition, you can do phenylchloroform extraction after the telomerase extension reaction. Some people do that all the time, whereby after the telomerase extension reaction, you perform a phenylchloroform extraction and then perform the PCR. And what that helps do is it reduces the background intensity of your trap products in the gel electrophoresis. So some people do this all the time, where they perform phenylchloroform extraction after the telomerase extension reaction. And then the other thing is you can check for the presence of inhibitors. So RNase inhibitors, for example, are very detrimental to trap. So you can check for the presence of inhibitors. You can check for the presence of inhibitors for the PCR reaction itself. For example, if you're using EDTA as opposed to EGTA, you want to make sure that you're not adding too much EDTA, that it's inhibiting the Taq polymerase activity. So these are things that you can keep in mind. And this concludes this module on trap.